Okay, so ladies and gentlemen, you are welcome. So in today's class, we are going to look at the concept of genome. Introduction. What is genome? A genome is a complete set of genetics material in an organism. So the genetic material, that is the information that are used in transmitting a characters from parent to the offspring, they are the genetic material. So a genome is the complete set of genetic material in an organism. So when you take the total of all genetic materials that are found in a living organism, so that is what we describe as genome. Like, for example, we have human genome, we have viral genome, we have bacterial genome. So we have different set of genomes. So each and every organism has their own genome, and it is the genome that make each and every living organism differ from one another. We are human. We have two legs, two eyes, two hands. We think, we imagine, all because of our genome. So genome is what makes one individual differ from one another. That is why if you check that in each and every living organism, they have their own specific number of chromosomes because the genome is the entire set of the chromosomes that are found in humans. So the genome contains an information that can produce a complete living organisms. Like we as human, we can from the human genome, we can actually get the complete human. We can get complete human. So, like for example, in human, we have 46 chromosomes. In Drosophila melanogaster, there are 6 chromosomes. In chimpanzee, they have 48 chromosomes. So what makes human differ from chimpanzee is just because of that two differences in terms of the number of the genome. And in addition, sorry, it's actually the difference in terms of the number of the chromosome. Like for example, in human, we have 46 and chimpanzees have 48. So the arrangement of the nucleotide or of the genome of human and that of the chimpanzee are different. And then the number of the chromosomes are also different. So as a result of that, we are actually differ from one another. So that's what makes us differ from one another. We have 46 chromosomes and they have 48. And in terms of this arrangement of the nucleotide sequence, they are also differ from one another. So that is why we are differ from them. If you check how horse, they have 65 chromosomes. So that is what I wanted us to understand. The number of chromosomes that are found in each and every living organism varies, and that is what makes us differ from one another. And of course, interestingly, the genome includes both genes and a non-coding sequence of DNA. So a genes is a segment of DNA, a code for a functional protein. So there are sequences of nucleotides that can express or that can produce a functional protein. While in non-codon region, there are regions that are not meant to produce a protein or to express a particular protein. So that is why they are called non-coding sequences of DNA. More interestingly, in humans, we have only 1.5% of our genome that are genes or that are functional or that are actually used to give or to generate a functional protein, while the remaining 98.5%, in the olden days, we call them junk DNA because they do not actually produce proteins. But recent advancement in technology we now found out that some of those non-coding sequences of the DNA, they are very important in actually identification of human life in the case of DNA fingerprint. Because with your own DNA, you can actually recognize a particular individual, especially like, for example, in the case of paternity disputes, in the case of committing any criminal offense, you can get the sample of the DNA and you can run it. So it's from those non-coding sequences that you can use to map the gene and actually fingerprint the human DNA. And from there, you can actually figure out the suspects. Also, in addition, these non-coding sequences, they are very important. They are very important in assisting the expressions of the genes. They are important in fact, some of them, they are regulatory elements that influences the expressions of the genes. So understanding genomes is crucial for the fields like genetics, medicine, and evolutionary biology. So if you understand the human genome, if you can get or if you understand the genome of a particular individual or of a particular patient, from there you can just easily actually treat a person. Like for example, in the personalized medicine. So in a personalized medicine is a type of a treatment where a patient's complete human DNA will be known, and then based on the genome, from there, then you can actually decide to put the patient in the treatment. So what I wanted to us to say, ladies and gentlemen, of course, in a cell, we have a cell, which is actually the structural and the functional unit of each and every living organism. And in these cells, we have nucleus. And this nucleus contains chromosome. So chromosomes have different components. We have centromere and we have telomere. A centromere is the center that actually joined the chromatids, while the telomere, they are found at the tail end of the chromosome. At the tail end, they are a repetitive sequence that are found at the tail end of the chromosomes, and that is what we call telomere. And the major importance of this telomere is to protect the DNA from wearing and tearing. So it is like a cover that trying to protect the DNA that is present in the chromosome. In fact, if you can maintain the integrity of the of the telomere, you can maintain the life, so you can actually prevent each in a particular patient. So I think, ladies and gentlemen, after this video, in this video, I'm going to attach the link for telomere as an anti-aging technology, that is reversing telomere as an anti-aging technology, because currently now, with the knowledge of the telomere technology, scientists are now using telomerase enzymes to reverse aging. So, ladies and gentlemen, after the chromosomes, as I said, as part of the chromosomes, you have centromere and you have telomere. So, of course, in the chromosomes is found with a histone protein which tightly packed the DNA. And of course, the DNA here is a double-stranded which contain thousands of genes. Like, for example, in humans, we have about 25,000 genes. Each of these genes is responsible for producing a specific type of protein. Okay, so then the next thing is actually the structure of the genome. So genomes are composed of DNA, which is structures at a double helix. So DNA is a double helix, So in eukaryotes, the genome is organized into chromosomes. So generally, in eukaryotic organisms, the genomes are organized into chromosomes located in the nucleus. While in the prokaryotic genomes, on the other hand, are usually circular and found in the cytoplasm. So this to tell you that remember in the prokaryotic cell, that doesn't have nucleus and membrane-bound organs. So that is why their genome is only, or their genomes are found in the cytoplasm and it is circular, while in the case of eukaryotes, it is double helix, the double stranded, which is double helix. So this is this. So you see here we have a DNA, and this is the histone protein that tightly wrap the DNA. And then, of course, the gene is the site of the DNA molecule. So this is a DNA that is found in the site of the genome. So there are repetitive sequences that are found in the human genome, which are responsible for expressing or for producing a specific type of protein. So then the Human Genome Projects. The Human Genome Project was actually an international research initiative completed in 2003. So the project is actually has actually started in 1990 and completed in the year 2003, which means that it took scientists 13 years to actually get the complete human genome project. So actually, because the human genome is very large, so the scientists have actually get the human DNA, break it into pieces and send it to different countries like US, France, Japan, China, and US to and Germany for them to actually get the complete sequences of the nucleotides that are found in the DNA. And of course, after completing the human genome, it successfully mapped the entire human genome, comprising over 3 billion base pairs. So that is why the base pairs that are found in actually human genome is that the nucleotides, there are over 300 billion. So this project has paved the way for advancement in personalized medicine and genetics research. There are so many diseases that actually scientists don't have idea on how the diseases are. So it's after they completing this human genome project that the scientists was able to map the human genome, and they were able to identify some differences in some nucleotides across individuals. And that is what even makes us differ from one another. Because even within human, we are not exactly the same. We don't look alike, but there are some differences. So it's through this human genome project that scientists was able to map why I why I as Muktar Ali differ from another person is through this. And one very interesting thing is that in the case of the patients that actually do not respond to treatment, so scientists can able to get their genome, sequence them, and from there you can actually develop a treatment that is specific for that particular individual. And that is what we call personalized medicine. And of course, genetic research, like sickle cell anemia, is after mapping the human genome project, the scientist was able to understand that SCLA is a genetic disease that actually occur as a result of mutation in one of the beta globin gene of hemoglobin. So we have different types of genome. First, we have nuclear genome, we have mitochondrial genome, we have viral genome. So that is why we have prokaryotic genome, eukaryotic genome, mitochondrial genome, and viral genome. So these are the four types of genome that we, ladies and gentlemen, and we shouldn't forget about them. We have nuclear DNA, mitochondrial DNA, we have viral DNA, and then we have prokaryotic DNA and eukaryotic. Although, if you check some other sources, that will tell you that we have eukaryotic genome, we have prokaryotic genome, we have viral genome, and we have mitochondrial genome. So, but what are the differences between these genome? So in the case of eukaryotic genome, that's where we're talking about the nuclear genome, because it's only in the eukaryotic that we have nucleus. So in the nucleus, we have genome, and it is double helix. It is a linear, we can call it linear, and it is double-stranded, and it contains both introns and exons. That is, they have both coding and non-coding regions. So that is for the eukaryotic. But for the prokaryotic genome, their genome is circular, and it doesn't have introns and exons, and they are localized in the cytoplasm. But in the case of mitochondrial DNA, it is circular. It looks like that of the prokaryotic genome. That is mitochondrial DNA and prokaryotic genome. They all have circular DNA. And that is why there is a theory called endosymbiotic relationship. Endosymbiotic relationship is a theory that tries to actually give an evidence that the bacteria that's found in human from evolutionary history, it actually came from bacteria. Some of the eukaryotic cells from the history, that when they engulf the bacteria, so the bacteria have actually transformed and changed into the mitochondria. And that is why if you check the mitochondria and, of course, the prokaryotic genome, to realize that there are a lot of similarity among them. There are a lot of. So that is actually the theory of endosymbiotic relationship, that the bacteria, the mitochondria that is found in prokaryotic cell came from the bacteria, which was actually engulfed from the history. So that is actually the theory of endosymbiotic relationship. It's a theory that's trying to demonstrate that the bacteria, the mitochondria of eukaryotic cell came from bacteria. So it's through the transformational changes of the prokaryotic bacteria that leads to the establishment of mitochondria in the eukaryotic cells. So that is why there are a lot of similarities between them. Then after the mitochondrial genome, we have viral genome. So viral genome can be single-stranded genome. It can be, because it's divided into two. We have either DNA viral or RNA viral. So in the case of DNA viral, can be double-stranded single RNA virus, double-stranded negative virus, or single-stranded RNA negative virus, or double-stranded RNA positive virus. So it's classified as positive, negative, double, and single. So nuclear genomes contain the majority of organism genetic material. That is one thing that you should understand. While the mitochondrial genomes are inherited maternally. So our mitochondrial DNA, we inherit them from our mother. So that is why if we want to actually, if you want to actually determine whose our mothers are, the best way actually is to map the mitochondrial. Like, for example, if you found, let's say there was an abandoned child and you want to know the mother of that child, how can you do that? You can actually get the suspects, the suspected mothers, get their mitochondrial genome, and get the mitochondrial genome of the child. Then from there, you try to see what we can do. So, mitochondrial genomes are maternally inherited, so you can map the genome of the child, sorry, the mitochondrial genome of a child and the mother, and from there you can actually see whether the child belongs to the mother or not. So that is one of the very interesting things with mitochondrial genome. And in addition, the mitochondrial genome is very important in the energy generation in the mitochondria. Remember that mitochondria is the powerhouse of the cells where energy are generated. So the genomes of the mitochondria is very important in actually generating energy of our cells. So what are the functions of the genome? So the genome includes instructions for building proteins that perform various functions. Like for example, the genomes will actually be converted into transcriptome, which is the RNA through the process called transcription. So a genome is the entire set of the genetic material of living organisms, while the transcriptome is the entire RNA that are found in the cell. Then translation, of course, the RNA will be translated into protein through the process called translation. So the proteins are called proteome. So the study of the entire set of the proteins in a particular living organism is called proteome. And of course, once you have a proteome, of course, you have activity. And after the activity, then you have the biochemistry of the cell. That is the function of the cell itself. And of course, cell is life, and cell is a structural and the functional unit of life. So you see, a full genome, it means the total amount of DNA of an organisms. So cellular genome, it can be haploid DNA with a content of the cell of an organism. So of course we have transcriptome, which is the total amount of RNA of an organisms, and then we have cellular transcriptome, that is the total amount of RNA of a cell of an organism. And then of course, we have full proteome, which is the total amount of proteins of an organism. And then we have cellular proteome, which is the total amount of proteins of a cell of an organism. Then, of course, for you to have a function, then you have metabolome, that is the total metabolites. Then lipome, that is the total lipids. Then we have phosphorylome, that is phospholipids. And then we have methylome, which is actually like methyl groups that are used in post-translational modification. So once you have that, then of course you have function. So regulatory element within the genome, there are actually a regulatory element, which is actually non-coding sequence of the genome. So they are actually important in controlling gene expression and cellular activity. So mutation in the genome can lead to genetic disorders or contributes to evolution. So genomics sequencing techniques. So various techniques exist for sequencing genomes, which include Sanger sequencing and next generation sequencing. So these are actually techniques that can use to sequence the genome of an entire organism. So you can use a Sanger sequencing, but it's very slow in the case of Sanger. So, and we have a next generation sequencing, which is actually very sophisticated. So the next generation sequencing allows for rapid and cost-effective sequencing of the entire genomes. So that is why now, because of this next generation sequencing, you can actually get the genome of a particular patient, because some people, you may find someone with a disease that is not known, that is very difficult to diagnose. So you can actually go for next generation sequencing. You can just get the genome, and then you map it completely, you sequence it entirely, and then you will now compare it with the normal human being. And from there, we'll know the differences. And after knowing the differences, we'll know exactly where there is the problem. And then you can actually exploit the patient for personalized medicine. So these techniques have revolutionized genomics, enabling large-scale studies and the discoveries. So since the actually development of this next generation sequencing and sequencing technology, it has actually revolutionized genomics, which enable scientists to actually come up with a lot of discoveries. So what are the applications of genomics? So genomics play a key role in personalized medicine, tailoring treatment to individual genetic profiles, which is the personalized medicine. And it is also vital in agriculture, helping to improve crop resilience and yield. So the knowledge of genomics, you can be able to improve crop in terms of the resistance to insects, in terms of the resistance to stress, and of course, you can also maximize yield of the crops with the help of the genomics approach. Additionally, genomics aids in conservation effort by tracking biodiversity and species health. So ethical consideration. The study of genomes raises significant ethical questions regarding the privacy and genetic data sharing. So issues such as genetic discrimination and informed consent are critical in genomics research. So societies must carefully navigate the ethical dilemmas to harness the benefits of genomics responsibility. Because by understanding the genome of one part of a particular individual, it means that you will have information of all that living organism. You will have information of all that human. But we need privacy and prevent also genetics sharing. But if there is informed consent and you actually give a consent to get your genome and fence it, then there is no problem. So that is why societies must actually be carefully navigate the ethical dilemmas to harness the benefit of the genomic responsibility. So what are the futures of genomics? Advancement in technology continue to enhance our understanding of genomes and their functions. So the integration of artificial intelligence is expected to accelerate discoveries in genomic research. So, of course, artificial intelligence is an intelligence system that tries to emulate human behavior. So the artificial intelligence incorporated into genomics research, it will accelerate the discovery. So the future explorations may lead to breakthrough in disease prevention and treatment strategies, because all we want is to actually try to come up with a process of reducing or preventing disease and also disease treatments. So in conclusion, genome is fundamental to life. So influencing trait health and evolution, ongoing research in genomics holds tremendous potential for improving human health and well-being. So as our knowledge expands, ethical and societal consideration will remain crucial in genomics advancement. So ladies and gentlemen, this is where we are going to stop for today's lecture. If you have any question, ask, and if you didn't subscribe and join my YouTube community, try to join for you to have access to all of our videos. Thank you very much.